Yeah, so thank you very much for the opportunity to present today. And yeah, it's been really interesting and insightful talk so far. So as Paolo said, I just wanted to introduce a little bit of a flavor of what we've got at the moment with 10xGenomics and sort of some of the trends that we're seeing in research, some of which we've already seen, and then just introduce kind of where we're going, both with the Visium platform and then ultimately just to touch upon where we're going with in situ as well. So for those of you not familiar, and I will move through this quite quickly, the Visium solution from 10x follows a fairly similar theme to when we do single cell analysis. So really what that means is we provide the reagents that you need to do, make a Visium library to look at spatial gene expression, and we also provide the software to do the analysis. So effectively the Visium system is composed of these following components. So one is the slides, which I'll explain in a moment. The rest of the kits that you need to make an Illumina ready library to sequence. And then once you've sequenced, you can pick up the outputs from the sequencer and you can run that through our pipelines to do the data analysis. And that portion is free, and you can just download the software to do those analysis for you. So the way when we just have a very sort of cursory look at how we're working with Visium, just over here. Then the way that this technology works, for those of you who are not familiar, this is a slide-based method. So we have a capture area. So on one slide, we have 4 capture areas. These are 6.5 by 6.5 millimeters. So you can mount a reasonable sized piece of tissue on there, and you're actually able to scan the gene expression across that whole 6.5 by 6.5 millimeters. So if we look at that in a little bit more sort of detail as we go through, then if we look at one of those capture areas, then what we have are 5,000 spatially barcoded spots arrayed in a uniform fashion across those. And then if we look at those spots in a little bit more detail again, then they're basically composed of millions of oligos. So each one of those discrete spots is built up of millions of oligos. And then what we do from this is we can use poly-DT to capture poly-A genes. So we're relatively species agnostic on this, so we can work with human, mouse, rats, fish, etc. So anything that's got polyadenylated RNA we can capture. And then essentially what we do is we do first strand-seed DNA synthesis. And the important point from this is we introduce into that first strand-seed DNA this spatial barcode in there as well. So ultimately, when we sequence and we deconvolute the data, what that's allowing us to do is take the genes that we've detected as being expressed using the spatial barcode back to a physical position on that slide. And from there, we can then build up the spatial expression across our tissue area that we've captured. If we flip that on its side and look at it just on the other axis, then effectively what we've got is, again, these very discretely arrayed spatial spots, and we're using color here to denote the differences in the spatial barcode. So we can mount our section on the slide, and then we can capture express genes and make a library from there. So fundamentally, that's how the technology works. And if we look at the kind of workflow that's associated with this, so just to say, there is no specific 10x equipment that's required for this at all. You just basically need access to the following pieces of equipment. So one is you have the ability to tissue section. Right now, we're talking about cryostat sectioning, but I'll give you an update on that as we move through the talk. So you do 10 micron sections or thereabouts. You mount those onto the Visium slide, you fix, you do a H&E stain, and then you image those on a fairly standard microscope platform. And then from there, you make a gene expression library, you sequence in the standard way using short read chemistry on Illumina, and then we can pick up the output file and run it through the 10x pipeline. So this has a dedicated pipeline for doing the data analysis. And the reason for that is obviously what we're doing ultimately is we're combining this H&E image along with the gene expression data. So we need to bring those two modalities together. And this is fundamentally what we see when we do this. So over here on the left-hand side, we have the H&E image. Over here on the right-hand side, we have the gene expression clusters. And this is rather than, or it's kind of similar, I suppose, in a way, but rather than being a cell, this is now looking at those spots representing one of those spatial spots in there as well. And then the software will bring those two data components together, and it will generate an image such as this. So we've got the H&E image and the gene expression data overlaid. And in terms of the gene expression, you know, if we look across sort of standard tissues, we can see sort of north of 5,000 genes when we do these kind of analysis. So even though this appears, you know, we're only maybe looking at the expression of individual genes, actually these different areas can be composed of the expression of hundreds or even thousands of genes in there as well. So it's a really quite a powerful technique to do a broad area of tissue and get the resolution down to hundreds or even thousands of genes when we do these kind of approaches. So in terms of some of the basic papers, I think we've got, I should have probably checked this before we started, I think there's about 80 or so publications right now with Visium, and I often use this one because it's just quite an easy sort of explanation of what people are doing. So this is basically asking the age-old question in Alzheimer's disease, that when we look at one of the tangles and the plaques, how do they actually exert any influence over the expression of the surrounding tissue? So using a mouse model on this and aging it between 3 and 18 months, and then effectively looking at the periphery of those plaques and looking at the normal tissue in there, then the question that the researchers are asking, you know, is there a perceptible change in gene expression? And if so, which genes are affected? So they've basically identified a series of genes that, yes, are changed by the cells being in the periphery to those plaques as compared to normal tissue. And they can follow those out over this 18-month period. And effectively what they're really identifying is all of the sort of standard genes that you would expect to be changed. So things like oxidative stress and those kind of markers, the genes that are involved in that are indeed changed. So in terms of a sort of simple overview of a publication, I think this makes quite a nice introduction to that as well. And they also actually run in situ as well in this as well for greater resolution ultimately. So one of the trends that we're really seeing at the moment, and actually we saw this in one of the previous talks when we were discussing intestinal development, is this notion of really doing a sort of high level pass or sort of denoting the different individual cell types using single cell, and then combining that data ultimately with the Visium data. So we've seen this now as definitely a pretty on trend area. So people are running single cell through the standard 10x workflows, and they're also combining ultimately the data with this as well. So 2 sort of modalities, Illumina sequencing again, and again you can do the analysis, or certainly the initial analysis using the 10x software. And then what we're seeing is various publications now are now calling out their own analysis pipelines where you can bring those 2 data sets together. So for example, we have things like Starfinder, Spotlight, and even some of the applications in Surat are able to do this. So at the moment, we see this to be a really pretty big trend. And just as an example of this, this is a paper that's still not fully published and is in bioarchives. But this again is looking at a fairly simple question, and it's really asking the question, you know, if we look at myocardial infarction, so apologies, this is quite a small figure, but if we look at that sort of central zone of the ischemic zone, and then we move out from that and we start to look at the gene expression around the periphery, you know, what changes can we observe? So this is actually a very detailed publication, and what they've done here is they've looked at the gene expression. They've also looked at the ataxi data to look at the epigenome as well, and the spatial data. And fundamentally what they're starting to do from this is they're starting to bring together these kind of biological processes that are involved in this damage that occurs in the ischemic zone, and that sort of scar phenotype that develops in terms of healing. So they've pulled out, they've called out various biological processes by combining the single cell RNA data and the attack data. And then what they've done on top of that is they've looked at the data from the Visium. So here you have on the left-hand side just a H&E image, and the arrows here are denoting the sort of fibrotic region within the heart, or the sort of scar phenotype. And then from there, they've kind of called out different cells and different features. So one is obviously what we can see is when we map back the fibroblasts, that they map very nicely back to that fibrotic region as you would expect. And what we also see around there is genes that are involved in the hypoxic pathway, we can see that there's a high level of expression in there as well. And then ultimately on the right-hand side, we can see the endothelial cells. So what we're really starting to do now is starting to understand the biology of the response to the damage in the heart, and we can see the endothelial cells migrating in terms of the wound healing. And then I think what's really nice from this, in terms of, again, looking at that sort of interplay between damaged tissue and between normal tissue, so just calling out this region here, what the authors have done from the single cell data is they've used that to map back over the spots the proportion of different cell types. So here in the damaged region, you can see that that's largely composed of different fibroblast components. And then as we move out again to the periphery, we can see that that's composed of different endothelial cell components. So I think as a snapshot of like a nice publication around combining different aspects both single cell and Visium, this is really quite nice. And also in the previous talk, we saw the intestinal development one as well, which I think pretty much shows the same kind of level of data as well. So that's a definite trend that we're seeing at the moment as we go through this. So just to change gears and sort of explain a little bit on where we're going at 10x. So I'm not calling out all of the applications, I don't think, but just some of the sort of perhaps more pertinent ones to your research. So the first one, which is available already, so this is the ability to now start to look at proteins, and we'll push more on this later in the year as well. So here we have at the top the sort of standard Visium with H&E stains. We can do very similar protocol as detailed on the bottom, so sectioning and then doing immunofluorescence effectively and imaging, and then overlaying the data as well. So that's the first kind of foray, I guess, that we've got at 10x into taking both gene expression data and protein data and starting to bring those data sets together. And this is supported within the software that we have now. So a very sort of simple overview of that. So if you look at the immunofluorescence data, and we're calling out specifically neurons here in mouse brain, so we can see the immunofluorescence image. We can also capture the Visium sort of gene expression when we follow the workflow through. And then what the software is capable of doing is bringing those two modalities together. So now we can see the immunofluorescence data. So particularly here, we see this sort of strong expression. And again, we can see that over here when we're overlaying the two data sets. And what we can now also see is the expression of genes in those different regions as well. And again, not to overplay this, but this is, you know, potentially hundreds or even thousands of genes that compose each one of these different colored areas within that brain section. One of the other avenues that we've gone down, and we do this both with single cell as well, is the notion of doing targeted gene expression. So most of the assays that we do are really whole transcriptome analysis. We have now the ability to make baits and do pull downs from our libraries. These are the common panels that we have. So cancer, human gene signatures just for annotating cell types, etc., neuroscience and immunology. So we have those panels and they're fully compatible with the Visium technology, and you can see that they're around about 1,000 to 1,200 genes. And really the big kind of advantage of this is really the sequencing that's required. So you can reduce your sort of sequencing component by about 80% when you do this. What we also now have is the ability to build custom panels. So in around about 2 weeks, we can turn this around. So we do just do the design on our website, and we can build panels of up to 1,500 genes, which you can go through and annotate yourself, and then we can send that away for synthesis, and then you can use those. And again, the big advantage of this is the sort of sequencing burden is dramatically reduced. So as you can imagine, when we're talking about doing fresh frozen material, one of the big pushes is the ability to take this whole workflow and do this on FFPE material. And this is something that we will have available in the next 1 to 2 months. So we're just finalizing this now. And this is just sort of showing the data that we get back from there. So if you look in mouse brain and we compare Visium for FFP with standard fresh frozen, the kind of read depth that we recommend doing around about 50,000 reads per spot. And here you can see in terms of total genes detected, in the standard fresh frozen Visium, we're just a little bit below 6,000, and we're maybe another couple of hundred below that when we do Visium for FFPE. So in terms of recovery of transcripts and in terms of annotation and identifying genes, we get really pretty consistent data between the two methods when we do that. Just a little bit more detail on that as well. So if we do similar experiment where we compare and contrast between FFPE and fresh frozen, and we do this in triple positive breast cancer. So we have FFPE on the y-axis and fresh frozen on the x-axis. When we do those correlations, we see really quite strong correlation between the two. This is obviously quite a bit more of a difficult experiment because effectively what you're doing is sectioning, taking half fresh and half for FFPE. So they're not quite the same sample type because they've been processed slightly differently. But again, if we compare within that breast cancer, the sort of sensitivity, again, we see really pretty consistent data between those as well. So we're really quite happy that this will be a pretty robust method as and when it comes out, which should be, as I say, in the next month or so. The other portion of this, again, is to take the H&E image, let a pathologist look at this and call out different areas in there. So fat, for example, and carcinoma in situ. So this is really a sort of freehand drawn pathologist image around the H&E image. And again, if we then take that through and we do the gene expression and the Visium, and then we start to overlay the data, we see a really quite striking concordance between the pathology annotated data, and the gene expression data as well. So we're really very happy that this will be a robust method. The next thing that we're moving to later on in the year is the ability to do multiplex protein analysis. So we'll be releasing this with the ability to do tens to hundreds of proteins, so gene expression, and overlay that with protein data as well. And again, that will be fully supported in the software, and this will be compatible with both FFPE and fresh frozen tissue. So if we just take a very sort of cursory look at the kind of data that we're seeing from there. So over here on the right, we have immunofluorescence, so CD29 and CD4, and we can see how that lights up across the tissue section here. Do a similar experiment, and for those of you familiar with the single cell, this is really an extension of what we call the feature barcoding technology. So essentially what we do is you take monoclonal antibodies and we covalently tag those with an oligo sequence, so we can build a barcode into those in terms of identifying which antibody that we're working to. We can sequence through that, and then we can deconvolute the data and overlay it as shown here. So we see the same kind of patterns that we see in immunofluorescence. So again, CD29 and CD4, and obviously we have that sort of pan gene expression data below that as well. So those are two modalities that we will be able to bring together as we go through this. Moving out a little bit further, we will move to Visium HD. So this will move away from the sort of standard Visium. So at the moment, those spatial spots are 55 microns in diameter, and we're looking at anywhere between capturing 2 to 10 cells on one of those spots. We'll increase massively the resolution on this, and this will take us to the single cell world. So the workflow will feel very much the same. Again, this will be compatible with fresh, frozen and FFPE material, but it will really now take us into the realms of sort of single cell resolution, if that's where you actually need to go for the sort of biological questions that you're answering. And then moving out a little bit further, and apologies, this is quite a flashy image, but we've also recently acquired 2 companies, so Katana and Recore, and we are currently working on those in R&D and really sort of bringing the approach that 10x have. So the ability to have kits to do this, instrumentation where necessary, and also support the data analysis. So this is just looking at 137 mRNA targets in FFPE melanoma. And again, this will now take you down to true subcellular resolution. So really, I'm kind of done there really. So to say thank you, James is here today as well, so we can take questions. But really what would hopefully giving you the impression of today is that, you know, we were building on the foundations that we have already with Visium, increasing the modalities that we can run to. And then what we'll do in the longer term is we'll kind of scale on that, and we'll increase the resolution with both the Visium products and also moving more into the in situ products. So I hope I've not overrun too much there. So thank you very much for your time today. And we'll be happy to take some questions. And please, if you have additional questions, both of our email addresses are on here as well. So feel free to drop us an email. So thank you very much.